Okay, so we are going to talk first about cell cycle regulation, and I'm going to share a whiteboard that I can draw on and use some pretty colors for you, so that will be, go. Yep. And once again, we have been told that we are good to go in terms of we aren't going to have any strange rainbow effects going on here. We're just going to be able to talk to you. Okay. So what this is, we are talking about cell cycle regulation. And anytime in biology, in this advanced college level and as you go on into biology, when you're talking about regulation, you're always talking about chemical signals that are meeting up with other chemical signals to tell other signals in a cascade flow of information and direction. So let's really quickly review in terms of just the cell cycle and what it looks like. So you know that it's a pie chart, right? And that the smallest slice of the pie, sorry, that's not completely in the center, but that small slice is the M phase when cell division happens. Right, and we're going this way. And then this is the G1 phase, the gap one, where you have growth of the cell and the cell is just doing its thing. Then you have the S phase, which is for synthesis and replication of DNA. And then you have the G2 phase, which is more growth and preparation for cell division, right? Okay, so we're always going in this direction. Now the other thing is that we haven't really talked about this much, but you know that there are checkpoints. There are checkpoints in these in these many places. Checkpoints I'm making in these red lines here. And today I'm just going to be talking about 3 checkpoints in particular. So we're going to call this one the G1 checkpoint. Okay. And we're going to call this obviously the G2 checkpoint. And we're going to call this the M checkpoint, I guess. We'll just call it that. Checkpoint, yes, Mrs. Matt can spell. All right, so these are moments in time where the cell is going to check itself to see if, in terms of the G1, I'm going to stick with the G1 checkpoint and just mainly talk about this one, just as an example. The G1 checkpoint is a moment in the cell's life when it is deciding whether or not to go on and commit to cell division. Because as soon as the cell goes into the S phase, it's going to be spending energy on multiplying its DNA and then going forward into growing big enough to go into cell division. So once you've left G1 and into the S phase, you're committed to dividing. So before you do that, there are certain things that the cell is going to check on. It's going to check that there's no errors in its DNA, right? So no errors in DNA. And you may remember this from the pear deck. The other thing it's going to want to check is that are there nutrients around? Are there enough nutrients to support division? So enough nutrients. And it will also check if the cell has grown large enough to have enough size, cytoplasm, and organelles to send to 2 new cells, right? So is it large enough? So these are all things that a cell that have to be true before the cell can be signaled chemically to move forward. Right? So these are the conditions that the cell might be looking for. Okay. All right. So now we need to talk about, well, there are chemical signals then that are going to zap the cell and tell it to move forward. Well, how exactly does that happen? All right. I'm going to choose a different color for this. Hopefully green will show up. So there are these chemical signals that are going to signal the cell to move forward in terms of the G1 phase to move into S, in terms of the G2 phase to move into M. In terms of the one that's in the M phase, it's literally checking that the metaphase plate has every chromosome correctly connected to the spindle fibers before anaphase goes, right? So that's what that one is. Okay, but we're going to stick with G1 and chemical signals in general, right? So the chem signals, a chemical signal I'm going to use simple language, tells the cell to proceed, right, into, in this case, into the S phase, which does mean that it's committing to division, right? Okay. So if that chemical signal is successfully there and successfully transfers its signal, the cell will start replicating its DNA and entering the S phase, right? If not, if any of these things here are not true or there's some problem, then something else might happen. Okay, so now let's go down and let's talk about these signals for a minute. So the chemical signals that, there's two types of chemical signals, okay? Two types of cell regulation, I'll say. Cell regulation signals. One is called a cyclin. There are cyclins, and I'll indicate those with a, I think I'll make those green circles. Let's say that that is a cyclin, all right? We're talking about a molecule here, so it's not like a bouncing green ball. It's a molecule, but I'm gonna represent it that way, okay? The other type is called a cyclin-dependent kinase. A cyclin-dependent kinase. They call these cdks. So when you see the letters cdk, you know what it's really talking about is a chemical signal. It's a protein, all right, that is dependent on the presence of particular cyclins. And because it's a kinase, you should know and you should remember that anything that is a kinase is a molecule that sticks phosphates on something. So the cyclin-dependent kinases are the molecules that are really doing the work. They are phosphorylating other molecules that are going to, in G1's case, stimulate molecules that are going to do the work of replicating DNA. And you stimulate a molecule to do work by phosphorylating it. So a cyclin-dependent, anything that's a kinase is something that phosphorylates something else. Okay? So I'm going to represent them. Let's just stick with Christmas colors here, with this symbol. And look, there's a spot there for that kinase to fit. Okay? All right. So here's the thing, and this should make sense to you because of the way I've drawn those symbols, those two molecules. A cyclin-dependent kinase cannot do its job unless it is bound to a cyclin. Okay? So a CDK, which is going to do the signaling of molecules to go into next phase, right? A CDK must be bound or connected, right? These molecules, remember, to a cyclin in order to function. Okay? In other words, it can't do any phosphorylating. It can't activate anything unless it looks like this. In terms of the things that I've drawn, the CDK is not going to be able to do any activating or any signaling unless it is in combination with a cyclin. There, it has to look like that. Okay, so that is a functioning signal molecule. All right, so how do they work? Let me just draw a picture of a cell. And let's pretend that that cell, okay, how do they work? All right, a CDK plus cyclin complex, right, activates worker molecules by phosphorylating them. Phosphorylating them. Example, let's say we've got a cell that's in G1, all right? So we have a cell that's in the G1 phase, right? There's its nucleus. Chromatin is all unstrung, right? And within that cell, let's say that we have, I'm just going to draw let's say that there are molecules, all right? Let's say that this molecule that I've drawn right here is a molecule that is able to go into the nucleus and start replicating DNA, right? But it is inert, it is inactive unless it's phosphorylated. So let's say that this is a DNA replicating molecule, or a molecule that's involved in that process, okay? It can't do anything unless it's phosphorylated. So along comes, you have cyclin-dependent kinases in there. And they are always there. They are constantly there, right? There's your CDK. But it's not bound to a cyclin yet, because cyclins, hence their name, their concentration fluctuates as each phase comes and goes. So the cyclins are only going to be present when what things are happening? Well, the cyclin only increases in concentration so long as the cell has checked that its DNA is perfect, that it's a big enough cell, that there is nutrients in the environment. As long as those things are present, along comes the CDK, binds to the along comes the cyclin, binds to the CDK. Now you have an activated CDK. So here is a CDK that is activated, right? It can do its thing because it's bound to its cyclin. So what does it do? It puts a phosphate onto this molecule. And now that's activated. And the cell now enters S phase. As soon as it has enough cyclins to activate the CDKs, which are going to phosphorylate those DNA replicating worker molecules, you can go into the S phase. So that's basically what's going on here. It's a simple way of explaining it, but that's basically it. There's one more thing I'm going to share with you. That's going to go down. Okay, that's fine. Okay, there's one more thing that I want to share with you, and that is the graph that you're going to see on this worksheet. So here's the question. There. How do these cyclin and CDK molecules fluctuate over time in terms of their concentrations, right? Within a cell, over time, you have the concentration of molecules that can change, right? Concentration of molecules can change over time. That's true of everything, of any kind of molecule, depending on what it is. So let's say that this period in time is G1, and then here's S, and then here later is G2, and then here is the M phase, right? So time going by on the x-axis. All right, so here's the thing. The CDK concentration stays pretty much the same no matter what phase you're in, right? The CDK concentration doesn't change. Those molecules that I had colored in red, right? These molecules here, those molecules, they're just always hanging around. Sometimes they're activated, sometimes they're not. It depends, hence the cyclin-dependent kinase term. It depends on the concentration of the cyclins. And cyclins, true to their name, cycle during the time of the cell's life. So now if I talk about, for example, the cyclin that's responsible for getting the cell into S phase would look something like this, and then go down to nothing again, right? So the cyclin, or the G1 cyclin, because each one's slightly different. The G1 cyclin, this guy, it fluctuates in concentration. So when it gets high enough for the to start binding to the CDK and making that funny red-green structure, it starts activating cells to activating molecules to get the cell to replicate its DNA. So remember that cyclins fluctuate, CDKs do not. Cyclins, their concentrations constantly change. Constantly change. CDK stays the same. Okay? Now that's my simplistic rendering of how this is all working. So now I want to, I'm gonna make this, okay? All right, so that's Mrs. Murdoch's explanation of how cell regulation is controlled by cyclins and cyclin-dependent kinases. I'm going to stop this recording now.